Hello class, this is chapter 4. This is the last section. It is going to cover part 6, 7, and 8, which are sort of the other organelles besides the endomembrane system. First, we will talk about peroxisomes. Peroxisomes are similar to lysosomes. They are modified vesicles. Instead of being acidic and having digestive enzymes, they contain enzymes and hydrogen peroxide. It's involved in detoxifying. It's also involved in allowing us to break down lipids for energy. So your peroxisomes are why you can use fat for energy. So they are pretty cool. You can often find peroxisomes hanging out with mitochondria, and sometimes a lipid droplet, which this might be one. And so it can pull fats, break them down for use by the mitochondria, and then the mitochondria can use those fats for energy, for making ATP. Alright, so vacuoles are usually a little bit larger than vesicles, and they are usually for either storage or digesting. So vacuoles often fuse with lysosomes to digest larger structures. Plants have a central vacuole and animals do not have a central vacuole. But animals will have regular vacuoles. The central vacuole functions to handle water pressure. Other vacuoles, again, are for storage or for degradation, for digestion. So again, I want to remind you that plant cells have a central vacuole, animal cells don't, but animal cells do have regular vacuoles. Regular vacuoles are a little bit smaller and they can vary in their function. Okay, so I don't know why it says central vacuole. These are the energy organelles. Your chloroplasts are only in your plant cells, but they can take sunlight and water and CO2, and they can make carbohydrates. Your mitochondria are in plants and animals, and your mitochondria can take carbohydrate and oxygen, and they can make energy, ATP. Chloroplasts have a double membrane on the outside, and this is one reason why we think it started off as a bacteria that was eaten by sort of the prototype for a eukaryotic cell, and then kept as a pet because it was useful because of this double membrane. It also has, chloroplasts also have their own DNA and ribosomes, and they replicate on their own cycle although it can be coordinated with the cell cycle. So if you look at a chloroplast as an outer membrane, an inner membrane, and then these thylakoid membranes. And each of these discs are a thylakoid. A stack of them is called a grana, or a granum. And they're green due to chlorophyll, which is the main synthetic pigment. For this chapter, just know that chloroplasts are in plant cells, and they are used for photosynthesis. We'll get into its structure a little bit more in other chapters, but you do want to know that this inner membrane is for the bacteria we ate, and the outer membrane is for the food vacuole that we put the bacteria in. Except we didn't digest it because, the plant cells didn't digest them because they found out they could do photosynthesis. Mitochondria are similar. They have their own ribosomes and DNA. They have a double membrane. And again, that's the evidence that they used to be bacteria that were eaten but then kept by the proto-eukaryotic cell. Mitochondria do aerobic respiration. They make ATP. So here's a little bit of their structure. They have this outer membrane, which is the food vacuole. And they have this wavy inner membrane, which looks a lot like aerobic bacteria. And so that structure is more evidence that they are sort of like pet bacteria. Anyway, just know the mitochondria may have once been bacteria, but their main function is making ATP. The cytoskeleton is way more prevalent in animal cells than plant cells, and that's because animal cells do not have a cell wall. And so the cytoskeleton takes over the function, some of the functions that the cell wall does in the plant cells and the bacteria cells, which is determining cell shape and giving some protection. Now, besides that, the cytoskeleton helps with intracellular transport, so stuff moving around inside the cell. So in that way, it's kind of like the cardiovascular system of the body. But it's also important for cell movement. So your white blood cells move sort of by crawling. And that pseudopod movement is regulated by the cytoskeleton. Lastly, the cytoskeleton is used for cell division. There are 3 types of cytoskeletal fibers. These are actin, intermediate, and microtubules. I would call these microfilaments, but the main one is actin. And then the rest of these names I will be consistent with. So microtubules and intermediate filaments. The smallest in diameter are the microfilaments. But they can be easily assembled and disassembled. So one of their uses is to pinch off the cell during cell division. The microtubules are the largest in diameter, but they're hollow, and they can also be assembled and disassembled easy. And they will guide the separation of your chromosomes during cell division. Your intermediate filaments do not assemble and disassemble easily. They are more structural for protection. When we look at intracellular transport, you need a couple things. You need a motor protein like kinesin, and you need microtubules. These motor proteins have feet that attach to the microtubule. And they have a receptor that will attach to the cargo, typically a vesicle. But you can have other cargo like mitochondria and stuff like that. The cool thing about these motor proteins, when they use ATP, they can switch conformation. And so the little feet will literally walk along the microtubule. And that will drive movement in a single direction. So that the vesicle moves, the motor protein moves, and not the microtubule. And here you can see images of these three types of filaments. So you can see microfilaments here in many different cells. Each pink dot there is a nucleus, and there's typically one nucleus per cell. So you can see how many cells are there. I can see a pretty well-defined outline of a cell here. And here, if that helps you visualize individual cells. This is one cell with an intermediate filament. The most common one is keratin, and keratin is what makes your skin strong as a barrier. And then there are three cells here. And the yellow stain is showing microtubules. And so they localize in different parts throughout the cell. The microfilaments are typically around the edges. They're strong around the edges. The microtubules are typically more localized around the nucleus. The microtubules are more for intracellular trafficking, but they also determine cell shape. And the actin filaments are more for cell shape and cell movement. And then the intermediate filaments are usually spread throughout, and they are more for strength or structure. Centrioles, these are one thing that animal cells have that plants don't. And they are in a certain area of the cell called the centrosome. And the centrosome and the centrioles control microtubule arrangement and structure and shape. And so they're basically for organization. They're really important in mitosis and meiosis when the cell divides. Cilia and flagella are projections. They project out from the cell membrane, and they're for movement. The flagella moves the cell, and the only human cells that have flagella are sperm. Cilia move. They move on the surface of the cell, and they will move some sort of liquid, usually mucus, but sometimes just tissue fluid. And so both of these move. They're both made from microtubules and motor proteins. But again, the flagella move the actual cell. Cilia do not move the cell. They move some sort of fluid, usually mucus. And if you look at cross sections of these, they typically are very geometric and kind of pretty. And that is it for our chapter. So let me know if you have any questions. Yeah, that's it.